Thank you, Ross, for the very kind introduction. And then first, thanks to organizers, especially Dan and Ross, for inviting me to attend this very exciting session and then have this opportunity to share with you some of our work. Also, thank you for waking up 1 hour early to attend this session. So I think we have this session because now many people are aware our society is facing a population aging with increased portion of senior individuals within the total population. So as we currently understand, aging is a universal process that affects the majority of multicellular organisms, including humans. So here are two self-portraits of Picasso when he was 25 and then when he was 90. So we can see after 65 years, the artist got more wrinkles on his face, but lose hairs. And aging does not simply change how we look outside. Intrinsically, it's accompanied with a decreased fitness at the whole body levels. So as different organs undergo age-related changes, we observe declines in functions and onset of diseases ranging from physical impairment, metabolic disorders, neurodegeneration, cancer, and more. So aging really is a single greatest risk factor for many different chronic conditions. So if we can target aging itself, we may be able to combat all those diseases at once rather than pursuing them one by one. Intriguingly, nature shows us this is possible. So if you ever met a centenarian in your life, you know what I mean. So those remarkable individuals do not simply live longer, they often remain healthy and active well into their late age. So while aging is universal, it does not affect all individuals in the same way. This is also true for our model system, Caenorhabditis elegans. So here I show you two videos of 18 days old worms, which is comparable in age to human around 80 years old. So looking at them, I think even if you haven't seen C. elegans before, it's not hard to tell the one on the right has aged better than the three on the left, and this one has also lived longer than the other three. So what causes difference? So why some individuals can live longer and enjoy a healthy late life? And actually, those are not new questions. More than 2,000 years ago, Aristotle already called an investigation into them. He asked, what are the reasons for some animals being long-lived and others short-lived? And considering the complexity of living organisms, these questions actually can be tackled from many different angles. And in our studies, we decided to approach this from the metabolic perspective, especially the regulatory role of metabolite. So metabolism is a complex network of intertwined chemical reactions. It's like a metro map. So at each stop, passengers get on and off as metabolite generated and flow through different metabolic pathways. The production of metabolite are highly organized in space across scales, from organelles to cells and to tissues. So over the year, we find that there are specific metabolites that can actively mediate the communications between different organelles within a cell, between different cells and tissues within organisms, and between different microbes and their hosts that within a shared ecosystem. And using C. elegans, we're able to decipher those metabolite-mediated regulatory mechanisms and review their importance in regulating longevity. So we primarily focus on two key sources of those single metabolites, the intracellular lysosomes and environmental bacteria. At the same time, as I mentioned, metabolism are highly organized in space. So we also want to review this spatial organization in vivo and to understand how it changes during aging. So we also made effort in developing new methods that allow us to image metabolism at a high resolution in vivo. So today, due to the interest of time, I will only focus on the lysosomes and two new methods that we recently developed. So lysosomes are highly specialized organelles that constitute acidic environment in eukaryotic system. And it contains around 60 types of hydrolases that operate at a low pH. And those enzymes can digest all kinds of materials delivered into these organelles through endocytosis, phagocytosis, and autophagy. And a diverse metabolite are generated from those processes. And one key class of lysosomal hydrolases are lipase, including LIPA. So its deficiency in humans causes Warman disease in infant and cholesterol ester storage disease in adult. And in both cases, severe lipid abnormality has been observed in livers. Several years ago, we have linked the lysosomal acid lipase with longevity regulation in C. elegans. So one is a homolog called lipo4, and we find it specifically expressed in intestinal cells. So worm has less fully differentiated tissues, so the intestine functionally resembled a combination of a liver, adipose tissue, and digestive tract. So my former graduate student, Andrew Follick, made transgenic strains to upregulate this lipase only in the intestine, and find this increased lysosomal lipolysis and also promote longevity at organism levels. He further uncovered that increased lysosomal lipolysis can activate a lysosome to nuclear retrograde signaling pathway. And this retrograde signaling is mediated by a specific fatty acid derivative called oleoacetylamine, OEA, and is facilitated by a lipid chaperone protein called LBP8. Here show you the structure of LBP8 solved by our collaborator Eric Altland's group at Emory University. So OEA can bind to this beta barrel pocket. So as shown in the model here, we find with increased lysosomal lipolysis, OEA generated at the lysosomes binds to the LBP8 proteins and transported by it from the lysosome to the nucleus, where it can serve as a ligand for the nuclear hormone receptors to activate transcriptional responses and promote mitochondrial beta oxidation. And importantly, all the components in this pathway are well conserved. And because C. elegans gene names can be confusing sometimes outside the field, so I list the name of their mammalian homolog at the bottom for your reference. And then we next want to know whether this lipid metabolite can promote longevity. So we have supplement OEA and its analog to wild-type worms and find this sufficient to prolong lifespan. And those OEA-treated worms are also more active at their later age. And then our collaborator, Hui Zheng's group at the Baylor College of Medicine has also supplemented those OEA analogs to their Alzheimer's disease mouse models and find the OEA treatment can attenuate A-beta-related pathology. As shown here, with OEA treatment, the A-beta plaque size and numbers are both reduced. There also lies APA1 positive microglia surrounding the A-beta plaques, suggesting reduced inflammation. Then in the water maze analysis, those OEA analog treated mice show improved cognitive functions. And furthermore, we also collaborate with organic chemist Meng Wang's group at Baylor and designed and synthesized a new OEA analog called MT30 to further increase its in vivo stability. And we find MT30 can improve metabolic performance in high-fat diet fit mice. So it's known those high-fat diet fit mice develop metabolic dysfunctions very similar to those observed in aged mice, including fatty liver and insulin resistance. So we find with just 5 weeks injection, MT30 can greatly reduce the lipid accumulation in the liver caused by high-fat diet feeding. It also improves insulin sensitivity and glucose tolerance. So together, it supports the beneficial role of this lipid metabolite OEA in promoting both lifespan and health span. And after finding the lipophore-induced lysosomal lipolysis can generate active metabolite signaling to the nucleus, we really hope to systematically understand how the lipophore induction can influence the lysosomal functions. So to this end, the former postdoc Yongyu, working together with two former graduate students, Youchen Guan and Max Gao, and developed a lysosomal immunoprecipitation method for adult worms. And this allows us to rapidly purify lysosomes and then use them to conduct both proteomics and lipodomics analysis using mass spec. Then we apply this method to compare the lysosome-enriched proteomes between wild-type and long-lived lipophore transgenic strains. And we find in wild-type conditions, there are normally around 200 proteins enriched at the lysosomes. And this number is more than doubled in the lipophore transgenic strains. And about 60% of them are unique to this long-lived condition. And this includes two nuclear porin proteins, NPP6 and NPP15. So in the heat map here, we compare the protein abundance between the non-lysosomal fractions and the lysosomal fractions. And as you can see, in wild-type worms, neither proteins show enrichment at lysosomal fractions. However, in the lipophore transgenic strains, both become highly enriched in the lysosomal fractions. So this observation puzzled us for a while because we pulled down lysosomal fractions, so why we can detect nuclear porin protein there, and these two nuclear porin proteins are the altering of the nuclear pore complex. So we thought about for a while until we noticed another candidate on the list, which is a DYLT1. So it's a worm homolog of a dynein light chain component. As you can see here, in wild-type conditions, it almost only detected in the non-lysosomal fractions, but it becomes highly enriched in the lysosomal fractions in the lipophore transgenic strains. So we know dynein mediate the retrograde transport of lysosomes towards the nucleus. So these findings, together the nuclear poring result, make us hypothesize maybe the interactions between the lysosomes and the nucleus increase with increasing lysosomal lipolysis. That's why when we pull down lysosomal fractions, we can also detect nuclear porin proteins. So to test this hypothesis, we image lysosomes in intestinal cells and find indeed there are more lysosomes accumulated around the nuclear regions in the lipophore transgenic strains. And then the quantification result also confirm the shift of the lysosomal distribution towards the paranuclear region. And we also acquired the HEM images at this high resolution. We further confirmed the increased proximity between lysosomes and the nucleus in the lipophore transgenic strains. And in some cases, we even see lysosome docking next to the nuclear pore. So this increased proximity between lysosome and nucleus may facilitate lysosome-derived signals into the nucleus and contributing to the longevity regulation. Consistently, when we knock down important component IPAS3 by RNAi to reduce the nuclear import, the lifespan extension caused by lipophore transgene is completely abolished. In contrast, when we knock down the exporting component, IPO1, to reduce the nuclear export, although itself shortened lifespan as the IPAS3 does, but it did not affect the lifespan extending effect caused by lipophore transgenes. So together, this really highlighted the importance of the lysosome-nuclear interactions in regulating longevity. So the increased proximity between these two organelles can facilitate the nuclear import of lysosome-derived signals and then help exert their effect on transcriptional controls and then contribute to the longevity effect. And in parallel, my former graduate student, Mazia Salvini, also used this lyso-IP method to compare the lipodomic profiles. and find the triglyceride species are decreased in the lysosomes from the lipophil transgenic strains, where the levels of polyunsaturated fatty acid, PUFAs, are increased. And she also conducted transcriptomics analysis and then find among the genes that are upregulated in the lipophil transgenic strains, the ones that involve neuropeptide signaling pathway are overrepresented. And this enrichment includes all the genes that encode neuropeptide processing and maturation, also over 30 neuropeptide encoding genes. Then we start wondering why the increased lysosomal lipolysis in the intestine can influence those neuronal genes. And Mazia find actually the polyunsaturated fatty acid PUFA are required. So here, she knocked down FAT3, encoding the FAT3 fatty acid desaturates to block PUFA biosynthesis. And then she measured the transcriptional levels of AGO3 and AGO21 to encode the neuropeptide processing enzymes. And you can see here, both genes are largely upregulated in the lipophil transgenic strains. But this induction are completely gone with a FAT3 mutant, suggesting PUFA is required. Then whether there are specific PUFAs involved in this regulation. So Mazia further supplement different PUFAs to the lipophil transgenic strains with a FAT3 mutant and find only DGLA, dihomogama-linolenic acid, is able to restore the induction of the either AGO3 or AGO21. Then how does DGLA generated from the intestinal lysosomal lipolysis signal to neurons? And Mazia find this is mediated by another lipid chaperone protein, LBP3. As you can see here, it is absent. The DGLA failed to restore the induction of either neuropeptide genes in the lipophil transgenic strains with a FAT3 mutant. So we also find that actually the LBP3 protein preferentially bind to the DGLA. So here the gray bar show you the percentage of PUFAs among the liposomes from the adult C. elegans. So you can see EPA take the largest portions, are close to 20%, and TGLA only take about 3%. However, if we profile the fatty acid that bind to the LBP3 proteins, now DGLA take the largest portions among all PUFAs. So together with the data I don't have to show you, don't have time to show you, we now find the increased lysosomal lipolysis can also generate lipid metabolite signaling to neurons in a cell autonomous manner. Then they act on neuronal nuclear hormone receptors to upregulate the neuropeptide signaling pathways. And the lipid metabolite involved in this cross-tissue communication is DGLA, and its effect is mediated by LBP3. Here, the structure of LBP3 solved by our collaborator, Eric, again, and actually we find it can switch between a ligand incompatible dimer and a ligand binding monomer. So we believe this switch between dimer and a monomer may help regulate DGLA binding and release, and then facilitate this neuron, the intestine neuron signaling. So now we have found both cell autonomous and cell non-autonomous mechanisms. We further show these two mechanisms actually act in parallel to promote longevity. As shown on the right, the induction of either LBP3 or LBP8 prolong lifespan, and their combined inductions produce an additive effect. So far, all the studies I show you occur in the parental generation where the lipase is upregulated. Last year, we published work revealing actually this longevity effect can be transmitted through multiple generations into genotypically wild-type progeny. And this finding was made by a former postdoc, Steve Zhang. So I show in the diagram, he crossed the long-lived lipophil transgenic strains with a wild type and then follow their wild type progenies over generations and then measure their lifespan and show in red dashed lines. And then same time, the wild type was also crossed with wild type and serve as controls, and their lifespan shown in the white dashed lines. So we found in the second and third generation, the wild type progeny from the lipophil transgenic strains live about 15% longer than wild type controls. And this lifespan extension starts to decrease in the fourth generation, that is great-great-grandchildren's. So when Steve first showed me this result, I was totally surprised because we hadn't realized that lysosomal metabolism can actively regulate transgenerational inheritance. So to understand the underlying molecular mechanisms, Steve looked for epigenetic factors that may contribute to this effect and screened a series H3 methylation marks. As shown here, he found that both H3K79 dimethylation and the trimethylation are higher in the lipophil transgenic strains. And using RNA-seq, he confirmed their genome occupancies are higher in the lipophil transgenic strains. And the similar induction was also observed in the third generation wild-type progeny. So the H3 methylation is known to play a very crucial role in epigenetic regulation, but K79 methylation is unique in several ways. So first, unlike other methylation sites in the tail region, the K79 site is located in the globule domain. And second, while other H3 methylation can happen at the free histone stage, the K79 methylation happens only after histone has been incorporated into the chromatin. And third, so far, no specific demethylase has been identified for K79. So it's believed this methylation can basically persistently stay for a very long time, is only removed when the entire histone is displayed during chromatin remodeling. So now we observe is upregulation by increased lysosomal lipolysis with persistence across transgenerational wild-type progeny. Does it contribute to the transgenerational longevity effect? So we first knocked down the K79 specific methyltransferase DAO1.3 in the second generation wild-type progeny and find this completely suppress the lifespan extension in the third generation wild-type progeny from the lipase transgenic strains. So suggesting it's required. Then how does it exert its effect? To answer this question, Steve integrated RNA-seq and the ChIP-seq result. And because it's known the H3K79 methylation is associated with transcription activation, so he searched for genes that are upregulated in the lipophil transgenic strains, and at the same time show increased enrichment of both H3K79 dimethylation and trimethylation. And this analysis helped him narrow down to 42 candidate genes. Among them, one called our eye. This is a H71 encoding H3.3 variant. So why is it interesting? Because it's known compared to canonical replication-dependent H3, the replication-independent H3.3 is more prone to be methylated at the K79 site. So we wondered whether the H71 and the K79 methylation can cooperate to regulate the transgenerational longevity. So we first applied inducible diagram approach to degrade H71 proteins only in the intestine in the second generation wild-type progeny and find this completely suppress the lifespan extension in the third generation, suggesting intentional H71 is required. Then whether it's sufficient. So Steve also generated a transgenic strains to upregulate H71 only in the intestine and find this can increase H3K79 trimethylation. And these animals are also long-lived, and the longevity effect can be transmitted through at least 3 generations. So suggesting the intentional H71 is also sufficient to promote transgenerational longevity. So now we know the histone H71 in the intestine is crucial. But for epigenetic transgenerational inheritance to occur, the germline is a key tissue because the epigenetic information must be transmitted from one generation to the next through the germline. So this raises a key question. So how does the intestinal His71 signal to the germline? Unexpectedly, we found the histone itself can be transported from the intestine to the germline. So how we made this finding? Because actually, Steve made the transgenic strains expressing flag tag His71 only in the intestine. Then he dissected the worm, conducted immunostaining. The goal is to validate the subcellular localization of His71. Then, total surprise, he finds in addition to the intestine, the flag signal was clearly detected in the oocyte of the germline. And this did not happen in the negative controls. And furthermore, when he knocked down RME2 to block the yolk uptake in the oocyte, and the flag His71 signal in the oocyte disappeared. So RME2 is a worm homologue of a VLDL receptor, and the yolk particles we know functionally resembled VLDL. So in C. elegans, it's synthesized in the intestine and then transported into the germline and uptaken into the oocyte through the RME2. So we further knock down all the yolk protein encoding genes in the germline, in the intestine, and then find the flag tag His71 signal also decrease in the oocyte. So together, it suggests this intestine-derived histone His71 piggyback onto yolk particles and transported from the intestine to the germline. And importantly, this transport is important for the transgenerational longevity effect because when we knock down RME2 to block this transport in the second generation, the lifespan extension in the third generation is completely gone. And we also find that DOT1.3, the H3K79 specific methyltransferase, only expressed in the germline. And when we upregulate its level only in the germline, it's sufficient to promote transgenerational longevity. So together, those findings highlight a previous unknown soma and germline epigenetic coordination mechanisms. So once the lysosome lipolysis is increased in the germline, the histone His71 is transcriptionally upregulated, transported into the germline by yolk proteins, where it's methylated at the K79 site by the DOT1.3 methyltransferase. But then how the His71 is transcriptionally upregulated in the intestine at the first place? So for these questions, we got very interesting answer when we look back to our lyso-IP proteomics analysis. On the list that are specifically enriched in the lysosome in the lipo4 transgenic strains, we find two AMPK catalytic subunit, AAK1 and AAK2. So in wild-type conditions, they are only detected in the non-lysosomal fractions, but become highly enriched in the lysosomal fractions. So suggesting with the increased lysosomal lipolysis, the AMPK might be recruited to the surface of the lysosomes. So Steve further generated a transgenic strains where the AMPK is constitutively tethered to the lysosomes only in the intestine. And he finds this will increase both H3K79 dimethylation and the trimethylation levels. And these animals are also long-lived, and the longevity effect can be transmitted through at least 3 generations. So I don't have time to show you all the data, but we also find this recruitment of AMPK to the lysosome will also displace mTORC1 from the lysosomes, reducing its lysosomal activation. At the result of this switch, the SKIN1 transcription factor is activated and then mediate the transcriptional upregulation of His71. And then this is what happened in the parental generation. Then in the subsequent generations, the elevated H3K79 levels will help sustain the transcriptional upregulation of His71, and then like cross generations and help maintain transgenerational inheritance. So overall, the activation of this lysosome to epigenome signaling pathway will lead to a H3.3 enriched epigenomes. And this enrichment will help improve genome stability and facilitate DNA damage repair, and thereby preserving a healthier epigenomic state to support both lifespan and health span extension. So together, our studies demonstrate that lysosomes as not just a scavenger center of the cells, actually the metabolic action of these organelles can directly generate signaling metabolite, can also dynamically influence signal transduction, and both contribute to the organelle and tissue communication and also organism longevity. But given the importance of lysosome metabolism, how to track the metabolic state of these organelles in vivo? This has been technically challenging with conventional fluorescence microscopy. To meet this challenge, we collaborate with Jinxin Chen's group at Boston University and develop box film. This stands for Optical Boxcar Enhanced Fluorescence Detected Infrared Photothermal Microscope. So it's a little bit mouthful, so just remember box film. And Jianpeng Ao, a visiting scholar, has been leading this effort. So before introducing this new microscopy system, I would like to ask you to think back to your college organic chemistry. So you may remember that IR absorption comes from the unique way chemical bonds vibrate. So each molecule has its unique IR absorption patterns. And based on these patterns, the IR spectrum can detect multiple metabolic groups at once, as shown here. However, because of long wavelengths of IR light, traditional IR absorption-based imaging cannot reach subcellular resolutions. Another challenge comes from the strong absorption by water. So this causes a huge background interference and make IR imaging in water-rich biological samples difficult. So to override those limitations, photothermal approach has been introduced to increase the spatial resolution. In the current design, we further applied optical boxcar configuration. So as illustrated here in this system, there's a one mid-IR lasers, and then that's focused on the samples through the reverse objective, while the visible lasers is directed from the top objective. And both lasers are modulated, but at different frequency. So in each cycle, one IR pulse is paired with two visible pulses. And during imaging, the IR laser can also scan a wide range of wavelengths. And once it matches with the vibrational energy of a chemical bond in the samples, the IR absorption occurs, and it will cause a local temperature increase. Then for the thermosensitive fluorescence probe nearby, this temperature increase will cause a transient decrease in the fluorescence intensity. Then when over time, the local temperature will return to the baseline, then the fluorescence intensity recovers. So basically the system is not directly measuring the IR absorption, but measuring a time-gated decrease in the fluorescence intensity caused by IR absorption. So in this way, the IR basically wavelengths encode the chemical specificity, while the spatial resolution of the system is defined by the visible laser, which is at the confocal levels. So this system helps us to suppress the water interference and mitigate photobleaching and also enhance signals. And we further incorporate AI-assisted data denoising and spectrum deconvolution, and to further enhance the signal-to-noise ratios and also resolve the composition of different molecules. Currently, we can confidently resolve these 8 metabolic groups. So with this holistic imaging and analysis pipeline, now for first time, we can map the chemical or the metabolic composition of individual organelles in vivo and in live organisms. So we first apply it to image lysosomes. So here show you one intestinal cells of C. elegans, and all the lysosomes are labeled with a lysosensor. So you can see they vary in their size and fluorescence intensity. But from a metabolic perspective, we don't know whether they are similar or different. And box film now make it possible. So in this example, highlighting 4 different lysosomes, you can see their spectrum show drastic difference. So this suggests even in the same cells, the lysosomes display striking heterogeneity in their metabolic composition. And this level of metabolic heterogeneity was not known before. And we further applied box film to analyze age-related changes in lysosomes, and then find in this heat map, all the IR wavelengths are plotted along the x-axis, and along the y-axis, each row represents 1 lysosome. So from this overview, you can see the metabolic composition of lysosomes change with age. So signal decrease in some spectral regions while increase in other regions, so suggesting a shift of metabolic content over time. And on the t-SNE map, you can see the day 2 lysosomes form a very distinct clusters, well separated from day 4, day 6, and day 10 lysosomes. And day 4 lysosomes also form a relative separate clusters with some overlap with day 6 and day 10 lysosome. So suggesting the shift of the molecular content began to emerge around day 4. And we further deconvolute the IR spectrum and then conducted quantitative analysis. And we found a series of micromolecules, including nucleic acid, ceramide, and glycogen, become accumulated during aging in the lysosomes. And then this suggests the lysosome degradation capacities decrease with increasing age as early as day 4. And consistently, when we extract signals from free fatty acid and triglyceride and calculate their ratios to assess the lysosomal lipolysis activities, we observe a profound decline start to emerge at day 4. And similarly, when we extract signal from free amino acid and proteins and use their ratios to analyze lysosomal proteolysis activities, it's also decreased. So together, it shows that the lysosomal dysfunction during aging occur much earlier than we expected by day 4, where animals are still very active and show no clear sign of functional declining at the whole body levels. So it tells us that lysosomal dysfunction is likely an early driver rather than a mere outcome of aging. This may explain why the longevity promoting effect was very robust and strong when we increase the lipase to boost the lysosomal lipolysis. And indeed, when we use box film to analyze both lysosomal lipolysis and the proteolysis activities, both are increased. And this method is not restricted to C. elegans. So here we have applied it to image the NPC1 knockout mammalian cells. So NPC1 is a causal gene for human Niemann-Pick type C disease. And we found when it's knockout, the lysosomes accumulate all kinds of macromolecules together with decreased lysosomal lipolysis and the proteolysis activities, which is consistent with the pathology of the disease. And then, in addition to lysosomes, recently we have successfully applied box film to image other organelles, including mitochondria, lipid droplet, peroxisome. In each cases, we observe previous unknown metabolic heterogeneity. So we are very excited by it, and our ultimate goal with these new imaging technologies is to map the metabolic state of diverse organelles across all cell types in a live organisms and to build what we call the chemical atlas of aging cells. So despite its power, box film only provides information at the level of molecular group. It cannot resolve individual metabolic molecules. But for profiling individual molecules, we know imaging mass spec is a very powerful method. In one scan, thousands of molecules can be identified. However, its spatial resolution is often not enough for single-cell analysis. So we have collaborated with Paul Timberg's group at Janelia and Linjun Li's group at the Wisconsin Medicine and develop a new tissue expansion method that is compatible with mass spec imaging of small molecules. So this method expands tissues without heating, denaturation, or digestion to better retain the small molecules in the samples during expansion. And we have named it TAMI. So in this method, the expansion factors will determine the fold of enhancement in the spatial resolution. So my former graduate student Lang Ding and postdoc Hua Zhang in Linjun's lab and Amy in Paul's lab first applied this method on mouse cerebellum because this tissue has this nice layered structure in its cortex. And here show you the result of a 2-phosphatidylcholine molecules without expansion. So you can see some pattern, but it's hard to clearly tell the 3 layers. And here's the result of TAMI with 2.5-fold expansion with enhanced spatial resolution. Now we can see these 2 PC molecules has a very different spatial distribution in the cerebellum 3 layers. And then this was surprising to us because our assume actually phospholipids are ubiquitously distributed across all cell membranes. But now we know this is not the case. Actually, for the majority phospholipid, we found they uniquely distributed in one of the 3 cerebellar layers. And then we can further expand the tissues by 3.5-fold. And then with 10-micron laser scan, now we can reach single-cell resolution. So individual Purkinje cells become visible. So now we can really see how the spatial distribution of PC molecules at the single-cell levels. And in addition to complex phospholipid, TAMI can also profile small metabolite, including OEA that we are interested, as well as small peptide. And then it can really offer an opportunity to do multi-omics profiling of intact tissues and revealing previously unknown metabolic heterogeneity. And in addition to brains, we have successfully applied it to many other tissues, and the list is growing when many people are trying this method. So we are using it to really look at the metabolic organization changes during aging. So to summarize, we really believe metabolite carries crucial signaling roles across the scales within the cells and mediate organelle crosstalk. At the organism levels, it signals from one tissue to another. On the temporal scales, it really affects how long animals live. And now with advancement in imaging technologies, we really hope to understand how the metabolic organization change with time, and then help us to identify new targets to promote healthy aging. So I'll end here. I want to thank all the current and former members, and then also our wonderful collaborators, and then HHMI to support our research. And thank you. I'm happy to take questions.